Hi, I'm Amy Marshall-Colón. I'm an associate professor at the University of Illinois at Urbana-Champaign in the Department of Plant Biology. And today I'm going to be telling you about how we are trying to increase crop production by connecting models from the micro to the macro scale. I'm also an associate editor for the journal In Silico Plants, which is an open access, peer-reviewed online journal dealing with all aspects of plant modeling. And also we publish software that is useful to the experimental community. So I hope that you consider publishing your work with us. During this DigiCrop conference, we're going to hear many different perspectives on what makes a digital plant. If you go shopping on the internet for a digital plant, you'll find this cute little pot that monitors soil, water, and temperature, and it'll even get angry with you if you've overwatered or underwatered the plant. But this smart pot isn't too far off from how some sectors view digital plants. The AeroFarms company has a smart digital system that allows for communication between the aeroponically grown plants and their growers to maximize growth while minimizing inputs. But another perspective on digital plants, which is what I'll be talking about today, is the in silico variety, where computer simulations are used to grow plants and make predictions about how they will respond to perturbations, whether they're environmental or genetic perturbations. But what both of these types of digital plants have in common is that the end goal is crop improvement. Most of us are aware that with climate change, it's going to become increasingly difficult to meet the global food demand for staple crops. So strategies are needed to improve food security, and many of these strategies focus on understanding the relationship between plant genotype and phenotype. And for decades, geneticists have been trying to understand the relationship between genotype and phenotype to design ideal plants or idiotypes. And over the years, we've gotten pretty good at breeding for idiotypes by taking into consideration mechanistic information and by making detailed observations about plant response to a changing environment. So this prescriptive idiotype breeding can be achieved by integrating new technologies like high throughput phenotyping, gene editing, and high performance computing. And one way to integrate these technologies in order to advance our understanding of genotype to phenotype relationships is through the use of mathematical modeling. So mathematical crop models let us do things in the dry lab that are difficult or even impossible to do in the wet lab, but they can help us identify new avenues for engineering or breeding efforts. So in silico experimentation and discovery allows users to mix and match plug and play modules to identify optimization strategies to improve crop traits, like testing out C3 versus C4 photosynthesis in a crop of interest, or maybe even swapping sorghum roots for maize to see if their deeper rooting profile will improve maize yield. So this component swapping requires a library of models that can be mixed and matched seamlessly to produce believable biological data. So a question that I had a few years ago was if we could leverage the thousands of mathematical models of plants that have been published over the last few decades to build virtual plants. And what I think would be most exciting in the generation of digital plants would be linking mechanistic models from the micro scale to physiological models at the macro scale. And as it turns out, this poses a number of problems. And so these thousands of models that have been published, they're often written in different programming languages, and they run on different chronological and computational timescales. And so what that means is that they can't directly communicate. There needs to be some sort of messaging system that translates the inputs and the outputs from these various models. And so that turned out to be more of a logistical problem than a biological problem. And this is where the Crops in Silico team came together. And we built this project around three components. The first is a computational framework. So that's the first piece that we need, is this messaging system to allow communication between these very diverse models. And so then, of course, the other component is the virtual crop models that we can treat as modules that we want to link together in this framework to build accurate representation of these crops. But to really do that, it has to be a community effort, since no individual lab has all of the models needed or all of the parameter files necessary to build these virtual crop plants. So these are the three components of the Crops in Silico consortium. And so first I'm going to tell you about the computational framework. And this was built by Dr. Megan Lang, who's a research scientist at the National Center for Supercomputing Applications. When we presented Megan with this problem of wanting to connect these very diverse models, she went to work and built an interface called vegdrasil that can overcome some of these logistical issues that we were facing. And it does so by coordinating asynchronous communication between models and parameter files. And it also automatically transforms data passed between the models. It performs this using parallel execution of the models. And this framework, vegdrasil, is open and available for anybody to use. You can access it from GitHub, and those websites are listed down here at the bottom left of the screen. So right now, Megan has built in capabilities where we can connect models that are written in Python, MATLAB, C, C++, R. She's adding support for Fortran and SBML right now. Next, I'll be discussing the virtual crop models that we're connecting using the framework. So we have published one proof of concept study where we asked the question, can we simulate soybean response to elevated CO2 at multiple scales? And the scales we're talking about here are at the level of gene expression, metabolic flux, which is really enzyme activities, and the macro scale, which is the leaf level photosynthesis. And this is the work of Dr. Yu Wang and Dr. Kavya Kannan, who are working with me and Steve Long at the University of Illinois. So like any good proof of concept question, we ask a question that we already know the answer to. And so studies at the University of Illinois soy face facility, and the face is for the free air carbon enrichment experiment, have shown that soybean has a marked physiological response to elevated CO2, specifically leaf photosynthesis increased 25% across growing seasons inside elevated CO2 rings compared to the same genotype of plants that were grown under ambient CO2 conditions. And this was met with a 16% increase in yield. And this yield response to elevated CO2 has also been observed in rice and wheat. Another important observation from soy face studies for soybean was that under elevated CO2, there is sometimes observed a decline in rubisco activity relative to the capacity for RUBP regeneration. So this has been observed in some genotypes of soybean under certain conditions. And what this means is that even though light saturated net leaf CO2 uptake, or what we call ASAT, increases, it doesn't achieve the theoretical maximum determined by the Farquhar model of photosynthesis. So a hypothesis that has come out of these observations from the soy face experiment is that re-optimized investment of resources away from rubisco and toward RUBP regeneration may increase photosynthetic efficiency and potentially yield. So to explore this question, we had to first build a mechanistic model of soybean photosynthesis before doing our scenario testing. And I don't have time to go through the details of each one of these models, but what I want you to get out of this is that the independent models describe the individual processes that they set out to do, and they do that pretty well. But our goal was to see if the integrated models can replicate biological phenomenon better than any individual model at a single scale. And so what this image is showing you are the pieces of information or the connection points between the models. We use gene expression data to calculate protein concentrations, which were used to update the metabolic flux model. Those newly determined fluxes were used to update the macro level model, and the macro model could also feed in environmental data down to the metabolic flux model. The key outcome of this model integration was that the integrated models were able to simulate field-observed photosynthetic response to elevated CO2. And what we're looking at here, so again, this is a standard ACI curve for carbon assimilation. If we integrate just the metabolic flux model with the macro level leaf photosynthesis model, we get a good ACI curve, but it's unable to predict carbon assimilation under elevated CO2, so it's blind to environmental concentrations of CO2 until we add the gene expression data. And now we're able to get this change in photosynthetic capacity under elevated CO2. And again, this is what was observed in the field from the soy face experiment for some genotypes of soybean grown under ambient, which is the solid line, and elevated CO2, which is the dashed line. So we now have this robust integrated model that we can now use for scenario testing. So if our real question is, how can we optimize photosynthesis, then we can go back to the individual models. And so at the level of metabolic flux, we performed a sensitivity analysis to identify the enzymes with the highest control coefficient. And so we identified several of them, and we can break them down to represent the different portions of the light and dark reactions of photosynthesis. So what you can see here is that we're breaking this into carboxylation, RUBP regeneration, and starch synthesis. We know that these enzymes, the genes that encode these enzymes, are regulated at the level of transcription. So then we went back to our gene expression data and built a gene regulatory network to identify transcription factors that are regulating these key enzymes that are controlling carboxylation, RUBP regeneration, and starch synthesis. From this analysis, we identified several TFs, but one in particular was GATA2. It's predicted to repress the expression of the gene that encodes RBCS and upregulate the genes that encode FBP aldolase and starch synthase 2. And what we can do with that is now we can perform in silico perturbations where we can overexpress or knock out the transcription factor and predict the changes in the expression level of its target genes. We then take these updated gene expression levels, feed it back into the integrated model, and so now we can make these macro level predictions of how crop physiology will change in response to this genetic perturbation. And what can be observed here from this simulation is that the overexpression of soybean GATA2, which is a homolog of Arabidopsis GATA5, we are able to overcome some of that limitation that's imposed by photosynthetic acclimation under elevated CO2, and we get an increase in photosynthetic capacity. So that's an example of some of the information that we can get using integrated models from the micro to the macro scale. And what we're looking to do now is to continue to scale up from the leaf level photosynthesis to canopy level photosynthesis. And we're doing this by using data-driven 3D visualizations, which depict the dynamic nature of crop canopies with spatial resolution. With this advanced visualization, we can see things in the data that we may have missed just by looking at numbers in graphs and tables. So for example, we can use 3D architecture to simulate light absorbance throughout a soybean canopy and feed that information to the photosynthesis model. We can then test and compare the light absorption in genotypes with different canopy architectures to get at that idiotype design. And we're doing this in collaboration with a co-PI on the grant, Dr. Bedrich Bennis at Purdue University, who in this example is using stress-based modeling on how wind can influence tree architecture over development. And really, what Bedrich has shown is that physics interactions can predict how the environment influences plant traits. So we now want to apply this type of modeling to our crop plants. And in fact, Bedrich is working with James Schnabel at Nebraska, and they have just recently published a paper using this data-driven visualization to make predictions about canopy light capture, and this time it's in sorghum. So Voxel, the title of the paper is that Voxel carving-based 3D reconstruction of sorghum identifies genetic determinants of radiation interception efficiency. And so what this is just really a unique way to use computer vision and procedural modeling to identify new plant traits that we were not able to measure in traditional ways, and then put those computer vision traits into a GWAS to identify genetic targets. And then another way that we're using the Yggdrasil framework to connect models is to connect independent canopy and root models to build a whole plant. And so this is a really great example of how we can use the dry bench to build these digital crops or these idiotypes by leveraging the best of both worlds, because there are really excellent root models like the OpenSimRoot model from Jonathan Lynch's group, and then also our canopy models of photosynthesis. And so with that, I want to thank my colleagues at the University of Illinois and the National Center for Supercomputing Application, and the other collaborators on the Crops in Silico project. I look forward to answering any questions that you might have. Thank you.